Hello. My name is Chandler Ross. I'm a PhD doctoral researcher at the University of Turku in Finland. It's lovely to be here with you all today, and I'm going to be talking about reconstructing stability landscapes, looking at microbiome data. So I have this table of contents slide sort of to keep me on track and to help just keep track of the presentation overall, because I'll be jumping between a few different topics and a few different areas. So to start off, I'm going to talk about what it is we're looking at, how we observe the dynamics, and what it means to look at microbiome data. So basically, we're measuring a subject's microbe abundances. That usually involves getting fecal samples and then basically taking apart all the DNA and counting all the different species that we find in there. And as you can imagine, there are practical constraints for this because you can only convince people to donate so many times, and other ethical constraints. So we end up in the end with a bunch of short time series consisting of a few time points each, or sometimes even just a single time point per individual. So this is what we're looking at in the figure. So we have many subjects often, but many short disjoint time series. And a lot of the models that work with this data, or want to work with, like a lot of dynamical models require continuous data. So this poses a bit of a problem. I also want to say that just the nature of the data we're looking at, in the same way that when we study gases, we have the ideal gas law, and we summarize all the different particles and their possible interactions, looking at macroscopic variables like pressure, volume, temperature. We need to do the same thing with microbial communities. So we have hundreds of species, maybe 200 to 300 in each person, and they're all interacting with each other. So let's say on the order of 90,000 possible interactions. And we need to break this down into something similar, and we're going to do the same thing and borrow the concepts from statistical physics and do some dimensionality reduction and talk about community components, which we refer to as enterosignatures in the literature. So it's a way of taking this community and breaking it down into something smaller that we can, more bite-sized chunks. So now that we understand the data and the problems, how can we infer these underlying dynamics? Now I'm going to talk about how I define stability and what it means in our context. So when you see time series data, like we have in the upper right, you can consider this from one perspective. The stability landscape perspective is moving along the landscape. So here we can clearly see there are two stable states, which are represented by the valleys, and they're separated. So we have alternative stable states. They're separated by an intermediate tipping region, so that's the unstable hill. And you can see, looking at the time series, it tends to spend time in one before hopping into another and back and forth. And these stable points are mediated by this deterministic landscape, but it gets nudged along through random fluctuations. So we have this interplay between stochastic and deterministic parts, and basically we want to disentangle them and see how they work together. I also want to point out that we expect stable states to shift. So here you can see some sort of reconstruction, or an illustration anyway, of what could be feasible or plausible, that we see the landscape changing with age. So we have the landscape as a function of abundance, or how much of a microbe there is. And we can see, according to this, in young adults, it'll have a low abundance stable state. As you get older, the stable state shifts and then breaks into two, and then in the elderly population we see one again. So these are the different things that we expect to see, and basically this is what we want to reconstruct. We want an entire 2D grid where we have the landscape as a function of age and abundance. So yes, new stable states can emerge and they move. Now, how do we model these dynamics? Well, we look at stochastic differential equation. So this, maybe many of you are familiar with this, that we think of x, our state variable, for us abundance, is going to be subject to a deterministic force, which is called the drift, which is related to the landscape, or it's a derived quantity from the drift, and then the stochastic diffusion part, g. And yeah, so we have the interplay between these two forces, and we need to learn f and g somehow. So the easiest way to go about that is to discretize the differential equation using the Euler-Maruyama approximation. So this is basically just first order. If you set delta W, well, if you set G to 0, then you would just retrieve the normal Euler approximation, first order approximation for ordinary differential equations. And this is the stochastic add-on. We can write down a likelihood given that expression. And here I have G is equal to e to the G hat, just so that when we're learning G, we can force it to be positive or non-negative because it's a variance, and that will help. And we have this delta T coming from the variance of the Wiener process. And as I mentioned, this landscape is derived from the drift quantity F, so we can compute that later down the line. The problem is we don't know what the drift or diffusion forms look like a priori, so they can be anything. We need something flexible to estimate them or to learn them. And the goal overall, as I mentioned, is to have this 2D landscape construction over age, where we're looking at the potential on the right. So here I have multiple snapshots. If I were to look at it from above, I would see this heat map of the stability changing throughout age. So essentially we have all the abundance ranges for the microbes, and we have the entire human lifespan is what we're trying to reconstruct for these enterosignatures, these microbial community summary components. Now comes the learning part. So I'm guessing most of you are familiar with Gaussian processes and their benefits and drawbacks. So they're really flexible. That's a great part since we don't know what these functions look like a priori. They're non-parametric. Again, good. We don't have any fixed form. We're not assuming they're cubic polynomials or quartic or anything like that. We can encode different prior assumptions. We can make them infinitely smooth. We can make them first order differentiable, second, and so on. And I use the infinitely smooth version with the exponentiated quadratic kernel. The main drawback being they scale as n cubed. So as the number of data points goes up, the computational time increases quite dramatically. In order to avoid that, we actually use the Hilbert space approximation. So we have F and G, the drift and diffusion, modeled by Gaussian processes, and this is the usual notation for it. You'll see X is bold because it's a vector over age and abundance, and K is our kernel, which is the covariance between the different points. So we have this grid where all the points have correlation between them. And the idea behind the Hilbert space approximation is we're taking the full covariance matrix K, breaking it down into a spectral density, and M star basis functions, which there's a lot of math involved here, and Laplacian operators and eigenvalues, but basically, yeah, we're approximating this full K with a much easier to calculate Hilbert space approximation. And the main benefit being that instead of scaling as n cubed, we have it scaling as the number of basis functions times the number of data points plus the number of basis functions, which is not an insignificant speed up. As I mentioned before, not only do we have short time series, but a lot of the subjects only have a single time point. So we have thousands and thousands of points, but they can't immediately be utilized in a dynamical model. So instead of discarding all this data, I want to find a way to include it. And you can see these are real histograms from some of the data I'm working with, where you can see it changes as in the different age groups of the population. In infants, we have very low abundance, and then their microbiomes build up, and we have much more higher relative abundance in children, and it changes for adults and the elderly. So it's really useful. There's information that we can extract from these stationary densities and build into the model. So the way I've been trying to do this is rewriting F, the drift, in terms of the diffusion and the stationary density, so reparametrizing it based on the Fokker-Planck equation. So we have this relation. And the good thing about Gaussian processes is that it now depends on the derivative of G, the diffusion as well. But we can sample the derivative directly just by taking the derivative of the Gaussian process, which is quite convenient. And then we can go back to our main model and say, okay, we have a zero mean Gaussian process. We can add a mean to that. That's the reparametrized version of the drift that we see above. And now we have all that stationary density information informing and basically pinning down the drift while we're inferring it. So the entire model overview is that we have cross-sectional data in the form of histograms. We can learn stationary densities from them. Then we plug this into the drift function. We have the diffusion. They're being learned from Gaussian processes. Plug them into our SDE model, where we input the longitudinal data, and then we have HMC, Bayesian inference, and Stan, and we get out our posteriors, F, G, and the hyperparameters for them, which are also being learned. So now the application case being a microbiome landscape, a little bit more detail about these enterosignatures. So as I mentioned there, every gut composition at every time point can be written as a combination of these different enterosignatures. So in the literature, they're usually pointed out as being 5. So you can say, for example, your 40% ES1, this subject on the left, 10% ES3, 30% ES2, and so on, up to 100%. And everyone will have a different composition, and yours will change over time too. So this is how we summarize the communities. And they're dynamic, but they haven't been proposed that long ago. So their stability properties haven't really been studied. So this is the gap we're trying to address. And if you want to know exactly where they come from, they're derived from NMF, so non-negative matrix factorization, where we have this count table of subjects for the rows and the microbes, or the species in the columns, and we're just breaking down into 2 submatrices with smaller dimensions that approximate this larger matrix. And now this is the preliminary result that we'd be looking at. So here's a reconstructed landscape over the ES fraction, so the relative fraction between 0 and 1, and the entire age span from 0 to 80 years old. And we can see the purple region is stable, the yellow region is unstable, and we have this age-dependent drift instability. We can see the stable state is moving with age, and we can calculate other things such as entropy or other markers of stability that are also changing with age. So those are the types of things we're interested in. So the key takeaways being short time series contain a lot of information about stability. We can input cross-sectional data into these models, and we have this workflow with Bayesian SDEs and Gaussian processes that allow us to flexibly reconstruct the stability landscapes. So with that, I want to thank my co-authors, Ville Laitinen, who is also here, my supervisor, one of my supervisors as well, and you can check out his talk tomorrow. But yes, that's about it.
Great, thank you so much for the fantastic talk. All right, do we have any questions? Yes.
So this is really cool stuff. Makes me excited to see what you can do also with the Hilbert space Gaussian processes. So I was just first trying to understand, so you had like 5 ES, so does it mean your X has dimensionality 5?
Yeah, 5 different enterosignatures. I've been looking at them individually so far, which isn't a great thing. I want to extend the model because, of course, they co-vary. They're relative, so if the percentage of one goes up, the other ones have to go down to compensate. But yeah, we have these 5 dimensions, or like 4, and then 1 degree of freedom.
Yeah, okay, makes sense. So follow-up question would be, of course, for the Hilbert space Gaussian processes, they have one bad scaling, which is the number of input dimensions, right? Because they're scaling, we have tensor products of spline. So I was wondering how you handle this, but you will still get to this, I suppose. When you have 4 input dimensions and you have to have the tensor product of all the basis functions.
Yeah, I haven't worked out how to make it scale exactly if I were to include all 5 of them at once, like this multivariate version. So far, I've had some luck and some results looking at them individually and just tracking the stability. But yeah, we have these degrees of freedom that could be a bit messy, but the work is still ongoing. But the Gaussian process, the Hilbert space approximations have been extremely helpful to even get this stuff going at all.
A small related question. Did you study the derivative HSGP in terms of how the accuracy of this is? Because we also have been doing derivative HSGPs in some places, and we noticed that the L we needed or M we needed to get kind of similar accuracy was different than from the non-derivative.
Sorry, the derivative for G?
We can also take this outside perhaps, yeah, later on if there's other questions.
This one?
Yeah, exactly, that the M we need is different from the M we need without the derivative to get the same kind of accuracy. And you may need to take care of the boundary factor. And I was wondering about your experiences, but we can also discuss this later.
Yeah, we can discuss it. So far, I'm using basically in the original paper, I think 2021, where they were giving suggestions for M star and the different length scales for the different dimensions.
Yeah, and I believe they may change with the derivative.
Yeah, using close to what they suggested in the paper. Haven't played too much with those dimensions, but it's given good results so far anyway.
Great. Any other questions? Okay. All right. Oh, sorry. Right there.
Oh. I see. I don't want to see the same hands all the time here. But. So have you thought about the difference between differentiating the approximation and approximating the derivative? We have this problem in STAN in that there's a lot of like CDF functions where no one's actually written a good approximation for the derivative, so we just differentiate through the algorithm. But in other cases, we know what the derivative is and can approximate that.
Yeah, I have thought about it. I just thought that one of the benefits of working with this framework is that we can sample directly from the derivative without taking the diffusion samples and then trying to approximate the derivative from them. So if we have the same beta random variable here, I don't know, maybe it's not reasonable, but at least that's how it first came to my mind.
And I had a comment as well, which is Simon's Foundation, which is where I work at Flatiron, which is part of Simon's Foundation, has a giant, it was founded as an autism research institute. And there's a group there that provides a huge amount, like $100 million a year in autism funding. But largely that goes to data collection, and one of their data collections is longitudinal gut biome for autistic children over long periods of time that's conjoined with medical records, RNA-seq data, so it's a very rich data set if you're looking for more.
Yeah,
more
always looking
places
for
to
more,
play.
of course. Thank you.
All right. I think that's everything. So let's give another round of applause to Chandler.